Absolutely. Here is a full word-for-word narrative script based on the 5 expanded sections above, written as a teleprompter copy. Every sentence is meant to be spoken aloud as written, with natural pauses and emphasis. Full teleprompter script. Dual coding and dopamine, your brain on oral reading. The journey begins the instant your eyes fixate on the first written symbol and your vocal folds prepare to oscillate. Reading aloud is not merely silent reading with an added audio track. It is a fundamentally different neurocognitive process that engages your brain as a highly integrated parallel processor. Within the first 30 seconds, your visual cortex, specifically the visual word form area in your left fusiform gyrus, rapidly decodes the orthographic features of each word. This region, sometimes called the brain's letterbox, has been honed through years of reading to recognize letter strings with astonishing speed, transforming abstract shapes into linguistic information. Simultaneously, the planum temporale and Heschl's gyrus, primary components of your auditory cortex, prepare to receive a very specific input, the sound of your own voice. This self-generated auditory feedback is critical because it triggers a process known as efference copy. Your motor cortex sends a predictive signal to your auditory system about what sound is about to be heard. When the actual sound matches the prediction, a powerful verification loop is established. This dual stream activation forms the basis of the phonological loop, a core component of Baddeley's model of working memory. The phonological loop consists of 2 subcomponents: the phonological store, which holds auditory information for a few seconds, and the articulatory control process, which rehearses that information subvocally. When you read aloud, the articulatory control process becomes overt, dramatically reinforcing the storage of verbal material. In just the first minute, your brain has effectively created 2 simultaneous memory representations, one visual and one auditory. Each is capable of cueing the other. This redundancy is not wasteful. It is your brain's most reliable strategy for ensuring information survives the rapid decay of short-term memory. Furthermore, the active vocal production engages your motor cortex, your premotor cortex, and your cerebellum, adding a third kinesthetic layer to the memory trace. Your cerebellum, long known for its role in fine motor coordination, also contributes to the timing and sequencing of speech sounds, embedding the rhythm of language into the neural representation. Consequently, reading aloud transforms a passive decoding task into an active, multi-sensory event. Each word becomes an action, not just a symbol. This is why individuals with reading difficulties, such as dyslexia, often show significant improvement when using oral reading strategies. The additional auditory and motor pathways can bypass or compensate for weaknesses in the visual phonological route. Your brain, ever the efficient survival machine, will use any available channel to extract meaning. By the end of the first full minute of oral reading, your visual, auditory, and motor cortices are firing in a tightly synchronized rhythm, a neural symphony that silent reading simply cannot conduct. As the second minute of oral reading commences, your brain moves beyond simple sensory processing and enters a state of enhanced cognitive integration centered on your left hemisphere's parasylvian language network. The superior temporal gyrus, which houses Wernicke's area, is now working in overdrive, not just receiving auditory input, but actively performing lexical semantic processing. It matches sound patterns to stored meanings. This region acts as your brain's dictionary, and when words are spoken aloud, the activation here is significantly more robust than during silent reading, a phenomenon consistently demonstrated by functional magnetic resonance imaging studies. Simultaneously, the inferior frontal gyrus, home to Broca's area, is no longer merely planning the motor sequences for speech. It is also performing a higher-order syntactic and grammatical analysis of the spoken stream. The arcuate fasciculus, a dense bundle of axons connecting Wernicke's and Broca's areas, becomes a high-speed data cable, transmitting information back and forth dozens of times per second. This dorsal stream pathway is the anatomical foundation of the phonological loop, and its engagement during oral reading is so intense that it can lead to measurable increases in functional connectivity within just a few minutes. A 2019 study using diffusion tensor imaging found that even brief periods of oral reading temporarily increased the fractional anisotropy of the arcuate fasciculus, indicating a more efficient transmission of neural signals. In essence, your brain physically reorganizes its white matter properties on the fly to accommodate the demands of overt speech production. Beyond the classic language areas, the angular gyrus, a cross-modal hub located at the junction of your temporal, parietal, and occipital lobes, plays a crucial role in binding the visual word form with its auditory and semantic representations. Think of the angular gyrus as your brain's librarian. It retrieves not just the meaning of a word, but also its associated sensory and emotional context. When you read the word ocean aloud, your angular gyrus may activate memories of salt spray, the sound of waves, or the feeling of cold water, enriching the bare lexical entry with a multidimensional experience. This binding process is the essence of semantic elaboration, a cognitive strategy known to dramatically improve long-term retention. Furthermore, the precuneus and the posterior cingulate cortex, core nodes of the default mode network, begin to modulate their activity. Normally, the default mode network is active during mind-wandering and internal thought, often distracting from externally focused tasks. However, the rhythmic, demanding nature of oral reading suppresses default mode network activity, reducing task-irrelevant thoughts and allowing your attention networks to operate with greater efficiency. This suppression explains why people often find reading aloud to be a powerful tool for focusing concentration when studying or working through complex material. By the end of the second minute, your brain has shifted from a reactive decoding mode to an active, integrative, and highly focused state, with multiple language and association areas working in concert. Entering the third minute, a remarkable neurochemical shift begins to unfold, driven not by the semantic content of the words, but by their rhythmic and prosodic structure. Your basal ganglia, a collection of subcortical nuclei including the caudate, putamen, and globus pallidus, are exquisitely sensitive to temporal patterns. Their evolutionary role in motor timing and sequence learning makes them ideally suited to detect the inherent rhythm of spoken language, the alternating stress patterns, the rising and falling intonations, the pauses between clauses. When you read aloud, your basal ganglia process this rhythmic information and, crucially, generate predictions about upcoming prosodic events. This predictive coding mechanism is directly linked to your brain's reward circuitry. The nucleus accumbens, often described as your brain's pleasure center, receives dense dopaminergic projections from the ventral tegmental area. When a predicted rhythmic pattern is successfully executed by your vocal apparatus, your ventral tegmental area releases a small pulse of dopamine into your nucleus accumbens. This is not the large phasic dopamine release associated with unexpected rewards like winning money. Rather, it is a tonic, sustained release that signals your brain this ongoing activity is valuable and should be continued. A 2017 experimental study from the University of Perugia directly measured striatal dopamine availability before and after a 5-minute oral reading task using positron emission tomography. The researchers found an 8 to 12% increase in dopamine binding potential in the caudate and putamen, an effect that was absent in a control group that read silently. This level of increase is comparable to the dopaminergic response elicited by listening to a favorite piece of music or engaging in a brief session of aerobic exercise. The implications are profound. Reading aloud is not a neutral cognitive act. It is a mildly rewarding one. Your brain learns to associate the act of vocal reading with a positive neurochemical state, which in turn increases your motivation to repeat the behavior. Furthermore, dopamine plays a critical role in synaptic plasticity, the cellular mechanism underlying learning and memory. Dopamine modulates the strength of connections between neurons by influencing long-term potentiation, particularly in your hippocampus and prefrontal cortex. When dopamine is present, the synapses that were active just before its release are stabilized and strengthened. Therefore, the dopamine pulses that occur during oral reading act as a chemical save button, tagging the just processed information for long-term storage. This is why information read aloud is not only better encoded initially, but also better consolidated into long-term memory. The third minute of oral reading is, from a neurochemical perspective, your brain's way of saying, "Pay attention. This matters. Remember this." You may experience this simply as a feeling of engagement or even mild enjoyment, but beneath the surface, a sophisticated neurochemical reinforcement system is actively reshaping your brain's learning priorities. As your reading session progresses into the fourth minute, the cumulative effects of dual coding, enhanced connectivity, and dopaminergic reinforcement begin to manifest in your brain's most evolutionarily advanced structure, your prefrontal cortex. This region, occupying the anterior part of your frontal lobes, is responsible for executive functions including sustained attention, inhibitory control, working memory manipulation, and goal-directed behavior. Reading aloud places unique demands on your dorsolateral prefrontal cortex, which acts as your brain's central executive. Unlike silent reading, where your attention can drift without immediate consequence, oral reading requires continuous output. Any lapse in attention results in an audible error, a stutter, or a loss of place. This real-time performance pressure forces your dorsolateral prefrontal cortex to maintain a state of vigilant focus. Neuroimaging studies have shown that the dorsolateral prefrontal cortex exhibits sustained activation during oral reading tasks, whereas silent reading often shows decreasing activation over time due to habituation and attentional drift. Simultaneously, your anterior cingulate cortex, a region involved in error detection and conflict monitoring, becomes highly active. Each time you self-correct a mispronounced word or adjust your phrasing for clarity, your anterior cingulate cortex generates an error-related negativity signal, a specific electrical potential measurable via electroencephalography. This signal, while momentarily uncomfortable, triggers a cascade of adaptive neural adjustments that improve your performance on subsequent trials. In essence, your brain is learning from its own vocal mistakes in real time, a process known as online error correction. Your ventrolateral prefrontal cortex also joins the network, serving as a semantic gatekeeper that selects relevant meanings and suppresses irrelevant ones. When you read a word with multiple meanings, such as the word bank meaning a financial institution versus the side of a river, your ventrolateral prefrontal cortex uses contextual information to inhibit the inappropriate interpretation while selecting the correct one. This inhibitory process is a form of cognitive control, and like any mental skill, it becomes more efficient with practice. Just 4 minutes of oral reading provides a concentrated workout for these executive function circuits, potentially leading to transient improvements in your cognitive flexibility and impulse control. Furthermore, your orbitofrontal cortex, which integrates sensory and emotional information to guide decision-making, receives input from the dopaminergic reward system. The mild pleasure associated with rhythmic oral reading may lead your orbitofrontal cortex to encode the entire activity as a positively valenced experience, increasing the likelihood that you will choose to repeat it in the future. This is the neural basis of habit formation. Your brain learns to predict that reading aloud leads to reward, and therefore prioritizes it over alternative activities that offer less immediate neurochemical reinforcement. By the end of the 4th minute, your prefrontal cortex is operating in a highly integrated mode, coordinating attention, error monitoring, semantic selection, and reward valuation simultaneously. In the 5th and final minute, your brain transitions from rapid, transient changes to the early stages of consolidation, a process that will continue long after your reading session has ended. Your hippocampus, a seahorse-shaped structure buried deep in your medial temporal lobe, begins to bind together the distributed neural representations established during the preceding minutes. This binding process, known as pattern completion, allows your hippocampus to act as an index that can later reactivate the entire memory engram from a partial cue. For example, hearing the same phrase read aloud days from now may trigger recall of the entire reading session, including not just the words but the auditory, motor, and emotional context in which you experience them. Your hippocampus is exquisitely sensitive to the dopaminergic signals emanating from your ventral tegmental area, and the dopamine released during oral reading directly enhances long-term potentiation within hippocampal circuits. This means that the 5th minute is not simply a continuation of the same activity. It is the point at which your brain begins to tag the entire preceding experience for offline processing during sleep and rest. Studies of memory consolidation have consistently shown that information is not stored immediately, but is gradually stabilized over hours and days. The neurochemical events of the 5th minute, including the sustained dopamine tone, the synchronized firing of hippocampal place cells, and the coordinated activity of your default mode network beginning to re-emerge, set the stage for this consolidation. Interestingly, the act of concluding your reading session also triggers a distinct neural event. The cessation of vocal motor output leads to a rebound in activity in your brain's salience network, including the anterior insula and the dorsal anterior cingulate cortex. This rebound serves to evaluate the just completed activity. Was it effortful? Was it rewarding? Was it worth doing again? Your insula, which represents interoceptive states, processes the subtle bodily feelings associated with reading aloud. The sensation of breath control, the proprioceptive feedback from your lips and tongue, the slight fatigue of sustained vocalization. This holistic evaluation is then communicated to your orbitofrontal cortex, which updates your subjective value of oral reading as a behavior. Over repeated sessions, this value signal strengthens, transforming reading aloud from a conscious cognitive strategy into an automatic preferred habit. Finally, even after your 5 minutes are complete, your brain continues to replay the recently read information in a compressed, temporally reversed order during subsequent quiet wakefulness. These events are known as hippocampal sharp wave ripples. They are essential for memory consolidation and are more numerous and more robust following active multisensory encoding tasks like oral reading than following passive tasks. Thus, the 5th minute is not an end but a beginning. Your brain's work has just started, and the seeds planted in those 300 seconds will continue to grow, branch, and strengthen, turning a brief moment of vocalization into a lasting neural treasure.